Hello, in this video we will discuss about the nephron. So the nephron anatomy and physiology. So first of all we should need to understand the kidney, that structure. So this is the basically is the adrenal gland which that is present in the form of pair. So here I am drawing just single, contain adrenal medulla and cortex, and here is the inside of the kidney contain a outer capsule, contain inside is the cortex and medulla is as a pyramid shape, and this is the pelvis, and this which that is excrete the urine and into the ureter to the bladder. So this is the structure of the kidney. Let's begin to understand the nephron. And where is the nephron? Nephron are present in the cortex and medulla of the kidney, which that is make up of the medulla contain loop of Henle of the nephron, while the cortex contain a glomerulus and Bowman capsule presence. So this is the glomerulus and Bowman capsule. Glomerulus is basically used for the filtration. So the filtrate will produce from the glomerulus. From the afferent arteriole move toward the Bowman capsule, while the efferent arteriole will move from the glomerulus to the to make a vasa recta for reabsorption of the waste, which that is not harmful but beneficial. And here is the proximal convoluted tubule, and this is the Bowman capsule, and this is the basically the loop of Henle, thick and thin loop of Henle. Thick is basically when end of the loop of Henle, while the thin will when beginning of the loop of Henle. So this is the distal convoluted tubule. This is a distal convoluted tubule will move toward the collecting duct, and this collecting duct is inside is contain a lumen. This is the basically lumen inside. So anyhow, so let's begin to understand the sympathetic stimulation, the activation of the sympathetic nervous system, which that will lead to vasodilation even. But the constriction of the efferent arteriole, the vasoconstriction will be occur. And in this way, the vasoconstriction due to the renin production. This renin which that is stimulated through, this is the precursor, renin is the precursor of angiotensin 2, which that will lead to constriction of the efferent arteriole, and efferent arteriole make vasa recta, and after the constriction that pressure will exert into the glomerulus due to the relaxation, the vasodilation of the afferent arteriole will lead to filtration. So that filtration is known as a filtrate. And this is the nephron. Nephron contain a glomerular filtration. Here let's begin to understand the glomerular filtration. So this is the afferent and efferent arteriole, which that will use for the excretion of the waste, and this is the glomerular filtration. But remember, during the waste filtration into the, this is the Bowman capsule, which that contain a Bowman urinary space, which that is the yellow color showing in that diagram, while this is the proximal convoluted tubule epithelium. So this is the urinary space. So when blood will move toward the glomerulus, toward the Bowman capsule, this is the juxtaglomerular cell, which that is responsible for the production of renin. So this is the glomerular basement membrane, which that is surrounded the mesangial cell. Inside is the known as the glomerular capillary. So this is the mesangial inside. But one more important thing, the red color showing that podocyte, and podocyte basically leave the food pores or food processes, which that will lead to increase the food for production of slit diaphragm. So that slit diaphragm, this is the glomerular basement membrane. Slit diaphragm basically is very important for containing a negative charge. So in this way, the protein also contain, amino acid also contain negative charge. Albumin protein contain negative charge, will not filter due to the repelling behavior. The repelling due to the negative charge are present in the slit diaphragm. And here the activation of the RAS, the renin-angiotensin system, will lead to intraglomerular formation of angiotensin 2, which the angiotensin 2 causes the constriction of the efferent arteriole, will lead to increase in the single nephron glomerular filtration rate. And in this way, if the more the problem can be arise, the proteinuria and the glucosuria due to the fibrosis and leading to the segmental glomerulosclerosis. And there is a lot of things we will discuss in the next video about the diabetic glucosuria. So let's begin to understand. This is the bigger picture of the capillary of the glomerulus, which contain innermost inside of glycocalyx. Glycocalyx is very important for the filtration of the macromolecule inhibition. So this will inhibit the macromolecule of the inside of the blood. So that is why the macromolecule will not filter. And on the other hand, it is also the regulator of the filtration rate. So on the other hand, here is you can see the this is the hydrostatic pressure within the Bowman's capsule, and this is the food processes through a podocyte which that produce food, which that produce the slit diaphragm, and this is the urinary space, and this is the basically is the hydrostatic pressure within the glomerular capillary. And hydrostatic pressure within the glomerular capillary, and this is the colloid osmotic pressure exerted by plasma protein retained within the glomerular capillary. So this is the hydrostatic pressure within the glomerular capillary, and this is the colloid osmotic pressure exerted by plasma protein retained within the glomerular capillary. And this basically is the plasma protein is responsible for the production of pressure, creation of pressure. But inside you should need to understand that is the hydrostatic pressure within the Bowman's capsule also. This is the hydrostatic pressure within the Bowman capsule. So when the hydrostatic pressure will increase in the Bowman capsule, reabsorption will be easy. So remember, why? Because the efferent arteriole will contain less water due to the absorption and filtration of the water also, and the water can be easily filter again reabsorb. So this is the pressure will exert inside, and in this way here is the hydrostatic pressure within the glomerular capillary will exert force to filtrate. So this is the equation shows that the glomerular filtration rate is equal to coefficient F into PC minus PB into a bracket to E minus pi C. Pi C is the basically is the hydrostatic pressure within the glomerular capillary. So in this way, let's here you can understand. This is the basically again I am drawing the bigger picture for understanding. Let's here is we will discuss now. So how it's possible? This is the glycocalyx, which that will able to hinder the macromolecule, but it will allow to move the potassium, sodium, calcium, and amino acid, fatty acid. On the other hand, there is a lot of micromolecule, for example glucose, and that will be absorbed and filtered as a filtrate. And this filtration will the albumin protein will not filter. Why? Because here is the slit diaphragm are present between the podocytes and podocyte food processes, which that will contain a negative charge, and inside the basically glomerular basement membrane contain negative charge will not allow to albumin protein move inside into the urinary space. So the urinary space. So in this way, the negative charge will negative repel due to the albumin contain negative charge, but the sodium, potassium, and calcium contain positive charge. Cation will move toward the negative charge of the podocyte slit diaphragm. So this is the podocyte processes, which that is the podocyte produce the food, and a lot of food will contain a slits. And this is the basically the normally I am drawing, but a lot of podocytes contain our food, produce a food. So this is the processes which that is very important for the retaining of the albumin inside of the cell and a lot of things. And glycocalysis is also important. Glycocalysis is basically also is the macromolecule. So this is the circulatory system. Let's begin to understand the from circulatory system, which that is provide the blood toward the afferent arteriole. So this is the afferent arteriole, and this is the efferent arteriole. This efferent arteriole make vasa recta. So vasa recta during the loop of Henle basically is used for the reabsorption of water, but here is the filtrate can be from the glomerulus to the Bowman capsule, sodium chloride, potassium, hydrogen bicarbonate, water, glucose, amino acid, creatine, and urea. But on the other hand, this is the proximal convoluted tubule, and reabsorption will be occur 65% sodium chloride, water, 100% glucose if the non-diabetic person. So the glucose urea will not occur. Amino acid and 90% the hydrogen bicarbonate will be reabsorbed. Why? Because if the excrete too much will lead to acidosis. So the secretion from the efferent arteriole toward the nephron can be the uric acid and organic acid. Well, let's begin to understand the countercurrent mechanism in the loop of Henle. This countercurrent mechanism means the vasa recta, which that is flow of blood, will be opposite, and the descending loop of Henle will move descendingly. The basically the increase of the constriction of the solute, while the decrease of the constriction of the solute will be opposite in the vasa recta. Let's begin to understand with the milliosmol per liter. So this is the isotope. This is the filtrate entering the descending limb becomes progressively more concentrated as it lose water. So this is the loop of Henle. This is the descending loop of Henle, which that is the thin. And this is the isotonic fluid will not move toward the water, will not move toward the vasa recta, toward the efferent arteriole, but when this is contains urea, water, potassium, sodium chloride, and it contains 300 when milliosmol per liter, but inside the vasa recta basically which that is surrounded the as a capillary efferent arteriole, which contain 500 milliosmol per liter, means the water content is less. So when the water content will be less, so the 300 milliosmol per liter content is more. So this is the basically is the blood in the vasa recta removes water and leaving the loop of Henle, leaving from the water leaving the loop of Henle. So water will leave to increase 600 milliosmol per liter. In this way, the 900 is the less water constriction, while the 600 contain high amount of 900 contain more constriction of solute, while the 600 contain less constriction of solute, will move toward the less constriction of the solute, and the water will easily absorb. While this is the 900 conversion. Why? Because the water will decrease, and inside the vasa recta should be 1200, and this is the opposite movement. And in this way, again, that is a 900 milliosmol per liter will move toward the lower constriction of the water. So in this way, the 1200 is the lower constriction of the water, but the solute will be more, will easily absorb. This is the easily absorb, and in this way the water can easily absorb, and it can be isotonic also. And again this 900, and remember that in this way the ascending loop of Henle basically is the thick loop of Henle, which that is potassium, sodium, and the filtrate becomes the hyposmotic thick ascending limb in this way. Here is the milliosmol mean the total solute concentration within a specific volume of a solvent expressed in osmol per milliliter. So this is basically the ascending loop of Henle, which that is reabsorbed the sodium, potassium chloride, move into the blood circulatory system in the vasa recta, will lead to increase again the solute concentration. When the solute concentration will be increased, it means the increase of the milliosmol per liter. For example, 900 to again 1200. So this is the countercurrent mechanism. In this way the sodium will absorb more, and in this way the sodium will absorb more but not water absorb, because the water is unable to absorb. In this way the increasing of the solute concentration will lead to increase, and this is the opposite direction. So this is the opposite direction. In this way you can understand. And when water will be reabsorbed, so in this way the increasing of the milliosmol per liter number mean the increasing of the solute, but the decreasing of the water will be sodium chloride and potassium will reabsorb. So in this way, let's begin to understand. This was the countercurrent mechanism, but this is the distal convoluted tubule. This is the distal convoluted tubule, which that is the aldosterone will produce an antidiuretic hormone through a pituitary gland release due to the low blood pressure for the increasing of the blood pressure. So the reabsorption of water, 5% sodium chloride and water, secretion of the potassium and proton into the nephron also, and the collecting duct which that reabsorb 5% water also and sodium chloride, and excretion can be the water, sodium chloride, potassium, hydrogen bicarbonate and creatine and urea, and still the water is 95%. So let's begin to understand the acid-base balance within the nephron, lumen and epithelium. For that is very important for throughout the blood pH regulation. So in this way epithelium, which that alkalosis if occur, in this way the acid decrease will lead to hydrogen bicarbonate ion combined with a proton will lead to produce the hydrogen carbonic acid. When carbonic acid will produce, it means the acid will increase, so the alkalosis will convert into the acidosis. But when increase the carbonic acid too much will lead to acidosis. So in this way the acid base balance, the pH should be the 7.35 to 4.5 should be maintained. So in this way, this is the carbonic acid can be dissociated through carbonic anhydrase to produce carbon dioxide, and carbon dioxide easily diffuse into the epithelial cell to the enter into the blood circulatory system and exhale. While if the acid base balance, in this way the alkalosis will be occur, so the acidosis should be require carbon dioxide. So thanks for watching. Please make sure to subscribe, like and share this video. Bye.